So joining us now, this is going to be the last session of our clinical trials arc for the day. And I think this is the one that many people are probably looking forward to the most. So we're going to hear from Dr. Mario Saporta, who's been involved in many clinical trials and is the lead actually for several clinical trials, several multi-site clinical trials. And of course, he's a professor of neurology and human genetics at the University of Miami Miller School of Medicine. He's also the medical director of the University of Miami CMT Center of Excellence, and he conducts both clinical but also translational research. He is the North American principal investigator for the FAR-Next Premier trial, and he leads a research laboratory focused on developing and applying human cellular models to the discovery and validation of genetic therapies and other drugs. Dr. Saporta, take it away.
Thank you very much, Keith. And actually, thank you for the CMTRF for inviting me to do this talk, which the topic is actually the best topic of, I think, the whole day. Not because I'm giving it, but because it's really a talk about really expectations and hope of what's to come. So I'm very excited to talk about that. Some disclosures. So I do work for consulting for biotech, so Swan Bio and FAR-Next. As Keith mentioned, I'm a PI for one of the clinical trials for CMT1A. I'm also the scientific advisory board for the CMT Association, and I receive funding from different patient foundations, the NIH and a biotech company, Swan Bio. So we are really kind of going through a paradigm shift currently over this last few years as neuromuscular specialists. We used to be the kind of Sherlock Holmes kind of figure looking for clues, trying to diagnose patients. And then as we saw this morning, we became molecular detectives looking for genes and trying to better diagnose patients. But this is now quickly translating into the development of different treatment strategies. And that there's a host of different options and tools that are being developed for different types of CMT, including small molecules or what we consider the kind of normal type of medications like aspirin or an antibiotic, but also genetic tools that are aimed to correct the primary genetic defect that's causing the CMT. And hopefully one day genome editing, as was discussed in the morning, as a more permanent option to correct the cause of CMT. So if we go back, and I think today was a nice day from a historical perspective to go through the whole process of the CMT research development over the last decades. If you think about CMT was first clinically defined in the late 1800s, so 1886. It took about a century to then find the first genetic cause of CMT, as we heard from the people that actually did it. And then it took about another 20 years to get to the first clinical trial for CMT using ascorbic acid or vitamin C. So from 2009 to 2013, the main publications of those different multicentric trials were published. Unfortunately, there were negative trials, and there are many reasons. I think that some of which were discussed this morning, those trials were negative, so we had to move on. And then it took 10 more years for another trial, also academic trial, to be published. And in 3 years, we saw the first industry-sponsored CMT trial with Acceleron and ACE-083, which was a phase 2 trial. Unfortunately, again, it did show some effect, but not functional improvement. And then only after currently, actually, so it's ongoing. We now have 2 trials actually ongoing and recruiting patients through different types of CMT. And we're going to talk a little bit about those trials. But if you think about how things are speeding up, from 91 to 2009, we identified dozens and dozens of CMT genes. And after that, with this knowledge and the understanding of the biology of some of the CMT subtypes, we are now way better organized to start thinking about how to treat them. And as Dr. Herman mentioned, we are also developing the tools to make sure that we design those trials correctly and that we also conduct those trials correctly so that we have the best chance to actually find a real effect and not miss it. And this is my favorite slide because I really think that the future is promising. So when I hit the button, we start seeing how busy this space is becoming. We have some of those names over there, but there are many names and some are not even in the slide, but those are all companies, either small biotech, larger pharma companies that are currently working in the CMT space. And that's unprecedented. We never had this environment happen, at least not since I started in this field. And for most people that have been around longer than I have, this is really something unique. And we are really hopeful that this will generate a momentum where success stories will start moving more and more players into this field. So if we start getting positive trials with the ones that are currently ongoing, this will provide a very strong proof of concept and a stimulus for other companies to become interested in CMT and start moving the field even further. And this is really needed because we know that there is a lot of work to identify potential treatments, and there's a lot of heartbreak in this process as well. So this is a typical slide that every drug discovery course will show, which is basically how much it costs for each phase of a clinical trial program, as was described by Melissa this morning, how much it takes to actually complete each phase, but also what's your probability of not completing or not transitioning from one phase to the other. So if you look, I'm going to try to point this here. So if you look at the initial work, which is finding good treatment candidates that you should invest your time and money to progress into treatments, it's pretty expensive to actually run those investigations and all the work that's required to identify those compounds. Probably half of them will not actually come into fruition to move into further study. They will show to be too toxic or not a viable target. So about half of them will be lost at that point. And then you're left with 50% of what you started. You're going to do your preclinical work, meaning testing into animals, seeing if you can show that the treatment actually works in animal models. And you're going to lose about 1/3 of those compounds. So you're seeing where we're going there. So we started with 100 compounds. We end up with about 51 here. And then probably at this point, we're getting to maybe a couple dozens of compounds. And then you go to phase 1 in humans. Again, about 1/3 of them will be lost. And this is another big area or problems. When you got to phase 2, you're looking now at patients. You want to see if the drug is safe in patients, if it has an efficacy signal. About half of your candidates will be lost there. And then when you get to phase 3, then your chances are a little bit better, but you're still going to lose about 1/3 of your candidates. And look at how much money has been spent so far in this development. And then by the time you're done, you may have 9 candidates out of the more than 100 that you started. So it's a cruel and very tough process. Of course, the better you get to this phase in terms of having a solid knowledge of biology, a solid lead, a mechanism that you understand, and you show that it's being affected by your treatment, the better you're going to be. And also in this phase, the better your trial is designed, the more likelihood you have to not fail because of lack of the ability of picking up a response. So this is called the value of death in translational research, because again, a lot of compounds will not go through that. Now, it's of course, it's daunting when you see that many different companies being on this space now to remain updated on what's going on. I know this was mentioned in the morning, but it's just to reemphasize that as patients, you do have a role in making sure that you don't miss an opportunity. So always work with your physician, but also always take a proactive step of going and look for potential opportunities to participate in research. Clinicaltrials.gov, it's still kind of the gold standard place to look for opportunities, just because every study that is being conducted in the US has to register in that site. So you're not going to miss anything because it has to be there. So if you just put Charcot-Marie-Tooth on the condition and hit search, you're going to see a bunch of studies. And then on the left side of the screen, you can actually select, okay, I just want to see clinical trials or interventional trials where something's given to the patient, or I'm okay looking to observational or natural history studies. So there are ways to make it more stringent, the results that you're going to get, so that you know what's going on and you can then not miss an opportunity. But again, always talk to your doctor. And if you find something that you're interested to know more, ask him to take a look for you and see if that makes sense for me, or see if even I'm a good candidate for this study. Because we are here to do that. So it's part of being a physician of a untreatable condition to always look for an opportunity for our patients to receive a treatment. So we're always going to be on the lookout for opportunities for you. Another way to make sure that you are updated on what's going on is going to a place that works with patients with CMT and have a large number of patients coming every year, because that will be the centers that will have the most updated information about CMT and about what's going on the research level. So this is a map of some of the CMT centers of excellence. This is from the CMT association, but the other associations also give this designation to different centers. So really, it's not, I'm not making any specific recommendation to where to look for them. But if you look on the internet for CMT Center of Excellence or some term around those lines, you're going to find places, hopefully near you, that you can go to be assessed by a CMT specialist and also have the opportunity to discuss research opportunities and potential clinical trials that may be ongoing. This is another way. There are also centers from the Inherited Neuropathy Consortium. I think you guys have been introduced to the INC now multiple times. This is a slide from Applied Therapeutics, which is doing the SWORD clinical trial. And what they did was basically go to the INC and then pick some centers within the INC to run the clinical trial, which makes sense because those are, the centers that are already ready to run those trials in terms of people trained, also all the different infrastructure that's required to run some of the evaluations. So if you just go, no, no, I think it's not the fire alarm. I think we're good. Right, don't evacuate, please stay. So again, it's so it's another way to look for places to be assessed is just going to the INC website and look for a INC center near you. So I'm going to just quickly review the two trials that are ongoing. So the first one is a trial for CMT1A by Farnext, and it's called the Premier trial. So this is a trial that's studying a small molecule. So it's a chemical compound, more traditional type of medication. It's in phase 3, meaning that it's really basically in the end of the process of validating this treatment. It's a 15-month study, and actually we're about to finish the first patients on this trial, the 15-month period, and start what's called an open label phase, where irrespective of whether the patient was taking an active medication or the placebo, everybody's moved to the active drug and continue to be followed, so that we can then see if the patients that were in the placebo can actually also improve, if there's an improvement in the treatment arm, and also see the longer term effect of the treatment. The evaluations are the typical things that we kind of consider for CMT, general neuro exam, nerve conduction studies, safety evaluations with EKG and blood work, a walking test, and measurements of muscle strength. It is a single dose of the medication versus placebo, 1-to-1. And the endpoints are the overall neuropathy limitation score, which is a scoring system for neuropathy, and the 10-meter walking test. The target population was of 350 patients, and we actually randomized 387 patients. And that was throughout 52 study locations, 25 in the US, 24 in Europe, and 3 in Israel. The recruitment is now closed, so we are basically now conducting the trial with this patient population. So we should be able to tell finally if this intervention worked or not. You can get more information again on that using that code on clinicaltrials.gov, or just looking CMT1A and FARNext. Those are the centers that are conducting the trial. So they're pretty spread out across the US, and also in Europe and in Israel. So we hope to have results of this trial probably next year, because the last patient completing the 15-month should be completing it around October next year. And then it takes some time for the data to be analyzed. And then if it's a positive trial, then probably around 2024, we should have the first treatment for CMT1A if it works. So we're very excited with it. If it works, it will be really the first treatment that we could offer our patients. Now, at the same time, we're starting a second clinical trial, which is amazing. We had none for decades, century, if you think about CMT as being first described in 1886. And now we have 2 trials happening. This one is for SORD neuropathy, which is a different type of CMT completely. In every aspect, it's very different from CMT1A. But it is probably the most common type of autosomal recessive CMT. And because of basically the biology of the disease, it lends really well to treatment with compounds that are already available and were developed originally for diabetes and diabetic neuropathy. So we're starting, or actually some centers have already started recruiting patients for the INSPIRE trial. That's the name of the trial for SORD neuropathy. It's testing a aldose reductase targeted inhibitor. So basically, with SORD, you have the loss of this enzyme that processes sorbitol. So sorbitol accumulates in the nerve cells, and that's likely what causes the nerve damage. And then by inhibiting the enzyme that's actually making the sorbitol, we hope to reverse the disease. So it's kind of a clean target, and hopefully we'll be able to show benefit. There's the email for patients that want to have more information about this trial, and I think it has been showing around on the screens also. And this is the kind of the timeline of the clinical trial. It's a 24-month trial, and that's really kind of the longest range of how long you can run a clinical trial, just because of how costly it is and how burdensome it can be to patients. It's going to really be the first trial using kind of the next generation tools for clinical trial design and performance, as described by David. So, so it's going to use the CMT functional outcome measure, muscle MRI, biomarkers to really try to capture a treatment response. And those are the centers. Again, most of those centers are either CMT centers of excellence or INC centers. So hopefully we're trying to keep the high quality of the data by using specialized centers. And the trial will likely be recruiting patients until the end of the year. It's of course it's a smaller population, so I think that the target is about 50 patients. But that's kind of the challenges with rare diseases is as you get down the CMT prevalence kind of scale, you start having very small numbers on specific subtypes of CMT, and designing the trials to validate therapies on those very ultra rare diseases are a challenge that we are currently focusing on. And then we have a bunch of therapies or treatment strategies that are now moving from preclinical studies near patient studies or clinical trials. So there's several new novel therapies using genetic intervention for CMT1A, including antisense oligonucleotide, shRNA, and genome editing. There's also gene replacement therapy, so what we consider gene therapy. One was presented today for CMT2, yesterday for CMT2S, but there's also interest in replacing mitofusion-1 for CMT2A as a treatment strategy. There's a lot of work now looking into how to target specifically the copy of the gene that has a mutation in autosomal dominant CMT, where patients can be only having one functional copy of the gene, and that could be the case for some of the CMT subtypes. And again, genome editing has a lot of promise, but a lot of work to be done. But there is now actually one company focusing on using a CRISPR inhibition strategy to treat CMT1A. So that's one of, at least as far as I know, one of the few efforts already using CRISPR for CMT that could be reaching the clinical kind of trial space very soon. And those are other treatments that are more, some of them are more generalizable in terms of having a broader effect in multiple types of CMTs, including HDAC6 inhibitors, which are small molecules, gene replacement therapy of a neurotrophic factor, a factor that stimulates nerves, and that has been shown to be, at least on preclinical work, helpful in multiple types of CMT, and it's soon going to be a clinical trial. There are specific small molecules for CMT2A that was presented yesterday, and also a modulation of certain cellular pathways for some of the demyelinating CMTs. So there's a lot of really good work, good biology, good candidates advancing through preclinical work that's coming very soon to clinical trials. And just as a quick acknowledgement, we say that it takes a village for clinical trials as well. This is our clinical coordinator, Danny, helping me carry boxes of supplies for our SWORD trial. But it's really our patients that we have to thank the most, because they are the ones that are really doing the hard work of coming to the visits, taking their medication correctly, being engaged, and helping feed this whole very kind of complex and expensive machine. So thank you for all of what you have done and what hopefully you'll be doing to develop this further. And that's kind of the initial part of this talk. And then I think we're going to move to the next one with Keith. But thank you.
Hi, Mario, it's Susan, and I'm going to give a little bit of a Q&A with you now virtually. I know Keith is there on stage with you, and you and I are just going to have a real quick discussion, which of course we intended to be in person, but alas, we've got this hybrid model. Yeah. So, you know, listening to Melissa earlier and to Dr. Herman, and now you, I really feel like I'm very excited about the pipeline and about the effort and the momentum that this community has. But I also want to help kind of distill all that information into really some things from a patient perspective. And one of the first questions that I think maybe going through a lot of people's minds here in the room and virtually is, you just named a bunch of different types of CMT. But if I didn't see my type of CMT represented, should I be discouraged that no one's working on my type, or should I be encouraged that at least CMT is getting addressed? I mean, how are the current clinical trials and maybe the planned clinical trials going to inform future clinical trials for different types of CMT?
Yeah, no, that's a great question, and yeah, there's no reason for being discouraged because we know this is a process. But every step of the way, we are not only learning from the trials that are being performed, but we are developing new tools that are applicable to every CMT type. So what we are measuring in most instances are things that apply to every CMT type. So how patients walk, how strong their muscles are, how their muscles are being affected by denervation on MRI, biomarkers. So all of these are things that can be applied to any CMT subtype. There's also this effort of trying to find pathways that might be common to most, if not every CMT. It's a very big challenge because the biology is so diverse, but it's possible that there will be specific targets that could potentially, if not cure, at least benefit most patients with CMT. So I think seeing that there's now interest of pharma, biotech to get into the CMT field, seeing that the tools that are needed to conduct trials have been developed and are being constantly improved, seeing that the mechanisms to recruit patients, diagnose patients, and bring them into research are also being further developed. All of this, it creates a ecosystem that will provide more and more opportunities for patients with different type of CMT to be in clinical studies and clinical trials. So I think it's a very exciting time for patients with any kind of CMT.
Thank you. So following that same thread, we all hear, the future of cures for CMT is in genetic medicine. And then we also have heard a number of times today, like if we could develop an aspirin for CMT, which of course is not a genetic medicine, that's a small molecule. So if I were interested in enrolling in a trial, if I enrolled in a small molecule trial, is that going to disqualify me for a genetic medicine trial or vice versa? How do I know as a patient how to choose which trials to enroll in, assuming I qualify for them all?
Yeah, so that's actually a very good problem to have, right? Having options of clinical trials to pick from. So but that's a real concern because we're talking about a rare disease, especially if you start dividing by subtype, it becomes ultra rare. Really depends on the strategy that's being tested. So for small molecules, usually after what we call a washout period, where patients are off the medication that was being tested, they are eligible for most trials after, and then that period is variable depending on how the trial is designed, but it can go from 6 months to a year. For the genetic interventions, they're a bit more complicated, especially if you're a part of a trial with a specific type of genetic therapy. For instance, if a virus is needed to give that therapy, that might disqualify you to receive another therapy with the same kind of virus because you can develop an immune reaction. So there are certain intricacies, and I think the best way is to really discuss that with your doctor, because it will be situational dependent. So each situation be a little bit different, but as a rule of thumb, if it's a small molecule trial, that will likely not affect your likelihood of being eligible for a future trial after a certain period of time. But depending on the gene-based therapy, it may affect your eligibility later on. So that's something that you need to consider.
Thank you so much. I have 1,000 more questions for you, but I don't want to monopolize all of your time. I think what we'd like to do now is kind of turn it over to the audience, both virtually and in person, to ask some of their questions for you.
Okay.
And I think, Keith, you're
Yeah.
going to lead that session, right?
That's right. Thank you. So thank you very much, Susan. And Dr. Saporta, if you'd like to go ahead and join me on the just seated on the stage here. We'd love to open it up now for questions from the audience here, both those of you who are online, as well as those of you who are with us in the room today. We do have one microphone here that's a standing microphone that you can make your way to. And we also have a runner who can bring a microphone to you. So any questions? What would you like to know about clinical trials? Well, I'm going to get us started because I have questions that I think other people probably share, but may not want to ask potentially. So what does a clinical trial look like from the perspective of a patient? What can they expect? So if they raise their hand and say, I want to be in a clinical trial, what does that really mean? What will they be doing?
So I think the first word that comes to mind is commitment, because you're going to commit your time, some of your lifestyle, because sometimes you do have to be more cognizant of taking the medications on the right time, filling out diaries, coming to clinic for the assessments on the specific dates that are set, because ideally we don't want to get data outside certain windows because everything is being done the same for the whole trial. So it is a big commitment. But at the same time, most patients really enjoy the experience because they feel like they're being extra taken care of, because we are in the trial, we're not only doing the trial itself and measuring the outcome of the trial, but we're always examining them. The other day I picked up a thyroid nodule on one of my patients, and probably his PCP missed that. And it was just because I was really being careful on examining and assessing the patient. So that there's that feeling of being kind of cared at a different level, but it comes at the expense of having to be sometimes traveling. We have patients that come from 3-4 hours away. A patient that comes flying from another state. So there is time commitment, there is lifestyle commitments, but I think that's basically it. Always ask when you're being interviewed to be part of a trial, what are the things that will be done to you, of course, because there are different things that trials would do. Some are more invasive, other are less invasive, and you need to understand what you are signing up to, because of course we can basically say, I don't want to be part of this study anymore at any point. It's your right to do so. But if you do, you basically took away the place of somebody that could have completed the trial. So it's very important to understand from the get-go what you're signing up to, so that you have a good chance that you're going to go through the whole trial and help provide an answer in regards to, is this a treatment that's worth investing and taking and using clinic.
So when you talk about a 2-year clinical trial, that is quite a commitment. But the person they're not staying in the hospital for 2 years, right? So what does that look like?
Yeah.
How frequently will they actually come to see you over those 2 years?
Sure. So for the Pharnex trial, it's a 15-month trial. Visits happen every 3 months. So every 3 months they have to be in clinic. And in between, every month they receive a call from the coordinator to check how they're doing, see if there's any adverse events happening, adverse effects from the treatment. But that's basically it. So it's about 5 visits in person. For the SWORD trial, it's a little bit different because they have frequent what we call telemedicine evaluations, where we connect with the patient and a nurse comes to the patient's home. So it's a home visit. And the actual clinic visits actually happen at the beginning, and then 12 months, and then 24 months. So there's a little bit more of a spread. But especially in the beginning, the visit, it's like a 3-day visit. So it's a longer visit. And that requires commitment as well, because you have everything paid for, the hotel, and so there's no expenses, but you have to separate those 3 days to travel or to be in the center, to go through the whole process of being evaluated. So yeah, you are not a prisoner for 2 years for sure.
And even the 3-day visit, it may at the beginning, that's not necessarily overnight. You might come.
Yeah,
get a
of
few
course.
things
You go
done
back
one
to your
day.
hotel, and then you come back in the morning, go back to a hotel, back in the morning, and then.
Great. So there's a question from the back.
So out of curiosity, because this is uncharted territory for CMTers around the globe, what would you consider success on a FARNEX trial? What is a clinician looking as far as success?
Yeah. So it really depends on how the placebo group will behave. If we can see the placebo group, it's declining throughout the course of the trial. If I see stabilization of the patients, that for me, it's a pretty good start. Of course, if I see the curves really kind of going opposite directions, that's the ideal situation. But 15 months, it's definitely not enough to expect to see significant improvement. And that's for, I think, every CMT trial, just because of how long it takes for your nervous system to improve, potentially regenerate or create mechanisms to recover function. So I would say if we see stabilization of the treatment arm and placebo going down, and then when we go to the open label phase, the placebo goes up and meets the treatment group that started earlier, I think that's a pretty good result.
So just another stupid question. That one of the measurable outcomes is it the 10-meter walk? What are you looking for there to get over there in half a second earlier, or
Yeah.
I mean, what's the purpose of that?
Yeah, that's a tricky thing because again, I would say that the FARNEX was still a old generation trial, and the SWORD trial, it's getting closer, though it's using similar endpoints. It's just looking at more things. It's a more new generation trial because it's using the newer tools that we have available. So the 10-meter walking test, nobody really loves it because it is a crude assessment. We were talking about that last night. There's a big learning effect for people, just regular people. If they keep being asked to walk fast 10 meters, they tend to do better over time just by training. But if the patient is getting worse, we should be able to see that. The question is whether they will get worse enough to affect their ability to walk 10 meters over those 15 months. So it's a hard question. But again, if we can see any stabilization of that time and patients not treated, getting more and more difficulty completing the task, it will be a positive test.
We do have one question from the audience, and I think this is another question from the audience. And I think this is the last one we have time for.
Hello again. A question about the endpoints for the clinical trial. Now, you may set out some objective measures for your patients to sort of meet. And let's say they don't meet them, but functionally they're a little bit better. You know, for some CMT patients, even a 5 to 10% improvement in their foot drop or some strength would make the world of a difference for them. Now, how do you anticipate, and that could be enough to not make the trial pass or get the approval. How do you anticipate bridging that, and what hope can we give our patients that, you know, functionally might be better, but it may not be enough to?
Yeah, so the
have a successful
difficult
trial?
the difficult thing is how you define that functionally better. Because so on a clinical trial, you have your endpoint, your primary endpoint, secondary endpoints, and then you have your exploratory endpoint. So you do have a bunch of stuff that's being made. If you see some signal or some of the secondary or exploratory endpoints, at least you have an argument to, okay, let's try to focus on this on a different program. From a trial perspective, you're still stuck to what because the whole trial was designed based on those endpoints. So you cannot change the rule of the game after the game is happening. So if you can show on any of those endpoints that this functional perception of improvement is real, at least there's a signal that would justify it, I think there's at least grounds to keep the program going. Now, if you have this subjective perception of improvement, but everything is negative or going the other way, you have to consider that there is an effect of being on a trial, as I mentioned, that might make patients feel better. And we cannot confound that with a real treatment effect, because then we'll be validating a treatment that's not going to help patients in the real world, where they are not going to be on a clinical trial environment and will not benefit from that kind of psychological or kind of potential benefit of being on a study. So that's kind of how we usually would think about interpreting the results of a trial and deciding whether it's worth continuing in that direction, or if, okay, it's a negative trial, we should just move on to something else.
All right, thank you very much. Please
Thank you.
join me in thanking Dr. Saporta.